Hello everyone, welcome to my channel Dr. Tapati's presentation. Myself Dr. Tapati Panjate. Today topic of the presentation is industrial uses of enzymes. According to Triven, the application of enzyme has been grouped into four broad categories, namely therapeutic uses, analytical uses, manipulative uses, and industrial uses. Already I have discussed about therapeutic, analytical, and manipulative uses of enzymes in three separate videos. You should watch those videos. Let's know about industrial uses of enzymes. Industrial biotechnology is gaining rapid grounds in the fields of industry and academic research, especially those of agriculture, food science, and medicine. This biotechnology field is mainly based on fermentation technology and biocatalysis. Due to recent advancements, biocatalysis using isolated enzymes is considered as economically beneficial than the use of whole cells. As you know that enzymes are biocatalysts produced by living cells to speed up specific biochemical reactions by lowering the activation energy barrier. They have been the center of attention for researchers, industrialists worldwide due to their wide range of physiological, analytical, and industrial applications. The main advantage of enzymes compared to most other catalysts is their stereo and regiospecificity as well as chemoselectivity. The practical application and industrial use of enzymes to accomplish certain reactions apart from the cell dates back many centuries and was practiced long before the nature or function of enzymes was understood. Industrial enzymes are the enzymes those are commercially used in a variety of industries. Enzymes and enzyme-based products are used in over 40 industrial sectors from household care and bioenergy to agriculture, animal health, and food. Enzymes are used today to make more than 700 commercial products. According to markets.com, the global industrial enzymes market size was valued at $6.9 billion in 2022 and is poised to grow from $7.4 billion in 2023 to $10.2 billion by 2028, growing at a CAGR of 6.6% in the forecast period 2023 to 2028. Enzymes are universally distributed throughout the animal, plant, and microbial kingdoms. Various enzymes are obtained from animal sources like amylases, lipase, renin, pepsin, trypsin, chymotrypsin, and catalase, whereas amylase, papain, glucanase, invertase, bromelain, phycin, and lipoxygenase enzymes are obtained from plant sources. Amylases, proteases, cellulases, xylanases, lactase, catalase, invertase, etc. enzymes are obtained from microorganisms. In spite of the wide distribution of enzymes, microbial sources are used for the industrial production due to various advantages. They are easily available and growth rate is faster. They are generally cheaper to produce. Their enzyme contents are more predictable and controllable. Reliable supplies of raw material of constant composition can more easily be arranged. Plant and animal tissues contain more potentially harmful materials than microbes, including phenolic compounds in case of plants, endogenous enzyme inhibitors, and proteases. Genetic changes using recombinant DNA technology can easily be done on microbial cells for elevated enzyme production and scientific development. Some of the main microbial sources of industrial enzymes include bacteria, fungi, and yeast. Most popular bacterial sources are Bacillus subtilis, Bacillus ligniformis, Bacillus acidodecubiens, and Bacillus stereothermophilus. Whereas if you consider about the fungi sources, those are Aspergillus oryzae, Aspergillus fumigatus, Aspergillus niger, Penicillium notatum, Trichoderma species, and Rhizopus species. Saccharomyces cerevisiae is the most popular yeast source for the production of microbial enzymes. Enzyme production is an expanding field of biotechnology. Although specific fermentation procedures adopted by manufacturers vary to a certain degree, there remain only two principal methods of cultivation: submerged fermentation or SMF and solid state fermentation or SSF. In case of submerged fermentation, the substrate is made soluble or suspended as fine particles in a large volume of water. Whereas in case of SSF, it is defined as fermentation process performed on a non-soluble material that acts both as physical support and source of nutrients in the absence of free flowing liquid. Enzymes are applicable in food material in various food industries, including starch processing industry, baking industry, dairy industry, animal and vegetable food making industry, and juice and wine industry. Similarly, enzymes are also applicable in non-food material in various other industries like leather industry, paper industry, detergent industry, and textile industry. Let's know about names of various industrial enzymes, their substrates, major functions, and valuable applications. First enzyme name is protease. Protease enzyme, in that case, substrates are protein and polypeptide. Protease enzyme catalyzes the hydrolysis of peptide bonds present in proteins and polypeptides. They are used in protease enzymes are used in laundry and dishwashing detergents industry, food processing industry, animal and feed additives in leather industry, waste treatment in case of pharmacology and drug manufacturer industry. Lipase enzyme, in that case, substrates are oil and fat. Lipase enzyme hydrolyzes the insoluble triacyl glycerols, catalyzing reverse reactions, as for example, esterification, alcoholysis, and acidolysis. Lipase enzyme is used in fat and oil industry, detergent industry, food and baking industry, pulp and paper industry, in fine chemicals industry. Whereas if you consider about the amylase enzyme, substrate is carbohydrate. It hydrolyzes starch into sugars. They are used in starch and fuel industry, baking industry, detergent industry, textile industry, pulp and paper industry, and in beverage industry. Pectinase enzyme, in that case, substrate is pectin. It catalyzes breakdown of pectin polysaccharide found in plant cell walls through hydrolysis, trans elimination, and de-esterification reactions. Trans elimination and de-esterification reactions. They are used in beverage industry, textile industry, food and animal feed industry. Substrate of xylane is enzyme is xylane. This enzyme involved in depolymerization of beta-1,4 glycoside linkage of xylane molecules into monomeric pentosane units, that is xylose. Xylanase enzyme is used in pulp and paper industry, baking and food industry, and animal feed industry. Whereas cellulase enzyme substrate is cellulose. It is involved in breakdown of cellulose found in plant cell walls into simple sugars as the glucose. Cellulase enzyme is involved in pulp and paper industry, food processing industry, animal feed industry, biofuel industry, and textile industry. Lacase enzyme, in that case, substrate is benzenediol. Lacase enzyme degrades lignin and oxidizes a variety of phenolic substrates, performing one electron oxidations leading to cross-linking. This enzyme is used in pulp and paper industry, textile industry, food industry, and bioremediation. Transglutaminase enzyme, in that case, substrate is protein or amine. It catalyzes the formation of covalent bond between gamma carboxyamide group of protein or peptide bound glutamine and the free amine group of protein or protein bound lysine. This enzyme is used in food and textile industry. Phytase enzyme, in that case substrate is phytate. It catalyzes the hydrolysis of phytic acid and releases a usable form of inorganic phosphorus. This enzyme is used in animal feed and food industries. Substrate of catalase enzyme is hydrogen peroxide. Catalase enzyme catalyzes the decomposition of hydrogen peroxide to oxygen and water. This enzyme is utilized in food processing industry, especially baking and dairy industry. It is also used in textile, pharmaceutical, and therapeutic, and it is also applicable in bioremediation. Glucose oxidase enzyme, in that case, substrate is beta-D glucose. It catalyzes the oxidation of beta-D glucose to D-glucono delta lactone and hydrogen peroxide in the presence of molecular oxygen. This enzyme is used in food industry, especially for the food preservation, flour processing, animal feeding, and in the beer industry. It is also applicable in animal disease prevention and control in medical diagnosis. Substrate of tannase enzyme is tannins. This enzyme catalyzes the hydrolysis of bonds present in the molecules of hydrolyzable tannins and gallic acid esters producing gallic acid. This enzyme is used in chemical industry, beverage industry, pharmaceutical industry, and in different food industries.